who is the CEO of GV20 Therapeutics, to speak about AI-based tumor target and antibody discovery.
Thank you. Hi, I want to thank Fair Journey and the organizing committee for inviting me to this meeting. Among all the speakers, I'm probably the least experienced with antibody discovery, so I'm really enjoying the learning process from all the audiences. By the way, GV20 is this acupuncture point on the top of the head, and we're trying to find cancer vulnerabilities. So it's been known over 20 years that HER2-positive breast cancer patients contain anti-HER2 antibodies in both their serum and tumor. So when people sequence the tumor, like RNA sequencing of a tumor, it's not just the cancer cells. Within the tumor, there are also immune cells infiltrating. And the T cells are making the T cell receptor against the tumor antigens, and the B cells are also producing the antibodies against the tumor antigens as well. And unfortunately, these B cells that are producing the antibodies are insufficient to really cure the tumor. And we hypothesize there are two reasons why this happened. One is that this tertiary lymphoid structure are not clonal expanded enough to produce enough antibodies to really eliminate the tumor. In addition, within the B cells, the cytokine environment can induce the B cell to shift the isotype from IgG and IgA. So from our own work as well as other published studies, we see that when the tumor is producing IgG antibody, it's more anti-tumor. But when it's shifted to an IgA antibody, the whole tumor microenvironment becomes more suppressive, and then it's more pro-tumor. And so then we thought, well, what if we just give the patients an abundant amount of the antibody with the correct FC? Then shouldn't we be able to develop these as therapeutics and cure the tumors? And that's kind of where we came into this problem. So nowadays, tumor RNA sequencing is really inexpensive. With $200 to $300, every lab or company can sequence a patient tumor. And the B cells that are infiltrating the tumor are also making the RNA that eventually translates into the antibody sequence. Unfortunately, the VDJ recombination creates a hypervariable sequence that's unmappable to the reference genome. Therefore, in the very first step of the RNA-seq analysis, we try to map the reads back to the genome to find where it comes from. In that first step, most of the antibodies get eliminated as unmappable junk. And so we have a computational algorithm that's already published. From each tumor RNA sequencing, we can de novo assemble those unmappable reads into antibody sequence. Because the framework sequence, there are some amino acid patterns. And so based on the sequence pattern, we know that they are coming from B cells, and we can assemble those into antibody sequence. So on average, from each tumor sequencing, like RNA-seq, we can get a few thousand antibodies. They are not all the antibodies in the tumor, but we've already compared the BCR sequencing versus the RNA-seq followed by this computational approach. We get the clonal expanded antibodies that are likely to be recognizing the tumor antigens. And so the first author of this paper then joined the company full-time, and we collected a big cohort of tumor profiles, and we have already extracted over 100 million tumor-infiltrating antibodies. And so then we were kind of thinking, what are these antibodies doing? How do we know the targets? It turns out we can use AI to in silico predict the binding targets of these antibodies without a prior training of a known antibody-antigen database. So it's a totally probabilistic approach. We are matching the 100 million antibodies with the 20,000 targets in the genome. And each target, we have a probabilistic mapping of, it's likely to bind to this target, but not the other target, and so on. The algorithm itself, we can't really patent this, so I can only explain to you intuitively how we do the mapping. So imagine for Google AI to learn how to recognize image from each other, right? So in order to know how to tell apart whether the picture is a pig, is a dog or a cat or tiger and lion, you need many, many labeled pictures. These are individually labeled pictures of one animal at a time. And if you have enough of those pictures, the AI can automatically learn the features to tell whether some new picture is a dog or a cat. And this, to us, is actually a fairly easy question because you have good training cases. But in our case, it's a much harder question. We are dealing with a tumor, with a mixture, right? So imagine instead of individual pictures, you are going to a zoo, but before you go to the zoo, you go to the website, say a London Zoo will say, we have the following animals, but supposedly they don't tell you how the animals look like. Instead, you can see all the pictures taken by tourists going to that zoo. It's a mixture of pictures. They don't really tell you that this is a tiger and that's a lion, but what you know is that these pictures are supposed to be coming from the animals in this zoo. And so if I only look at the pictures and animals in London Zoo, I can't really match them. However, if I have a lot of zoos and the zoos are all slightly different, then I can use AI. Intuitively, you can think, okay, let's look at all the zoos that have tigers versus the zoos that don't have tigers. What pictures do they have in common, right? Like to say these are tiger pictures. At the same time, we also need to look at all the zoos with lions. What do they have in common that are absent in the zoos that don't have lions? And so on for like giraffe, for hippo, for any animals, all of them together. And if we have enough zoos, we can actually do a pretty good training. And how do we know we are learning this correctly? Imagine we leave some zoos out. Imagine this is Zimbabwe Zoo. It doesn't come into the training, but after we finish the training, we look at the pictures in Zimbabwe Zoo, and if our AI can say, well, based on the pictures, they have a giraffe, hippo, but they don't have tiger or lion, and it's correct, if it matches with the animals in the zoo, then we know our AI is training correctly, right? What does it really learn? It is actually learning the features that can tell one animal apart from another animal. And you can see in this situation, we don't need to know how animals look like ahead of time. It's completely learned de novo from the collection of many, many different zoos. And so in our situation, every tumor is like a zoo. The antigens in the tumor is like the animals in the zoo. And the antibodies in each tumor are like the pictures in each zoo. And so we can simultaneously infer all the antigen-antibody pairs de novo without information on any existing antibodies against the known targets. And so the AI is automatically learning some peptide features instead of the full-length antibody sequence that are repeatedly expanded in many tumors with the target antigens. It's like when you try to really tell one animal from another, you are not relying on the whole picture. It's learning some important features like a patch on the body or the eye feature, and that alone you can tell this is a tiger, right? So when we are learning the antibody sequence, it's not the full-length sequence. Even partial sequences, some like 6 amino acid peptides can be important. And the AI can automatically figure out how long and what the features are. And so that's the beauty of using AI. So basically, we can use this to actually get antibodies. This is just one example. There are good anti-PD-1 antibodies in the patient tumors. We can build an AI model against PD-1 without knowing any existing PD-1 antibody sequences. And with that model, for every PD-1 antibody, given the sequence, we could see whether this is a PD-1 antibody. And so there was a paper where they synthesized the 10 clinical PD-1 antibodies. And we string each of these through the AI model. And interestingly, we found that our AI predicted binding actually even agrees with the measured Beer-core affinity of the PD-1 antibodies. And you can see the earlier PD-1 antibodies from Bristol Myers Squibb and Merck actually have lower affinity. The later ones from Novartis and Pfizer, and also one from Beijing, has much higher affinity. And with this, we can use AI to get an even higher affinity PD-1 antibody. And it has really excellent blocking ability to block PD-1, PD-L1 interaction. And yet it has a different binding epitope from Keytruda. So it doesn't compete with Keytruda. We tested this antibody in B16 model with a humanized PD-1, and it actually has very good in vivo efficacy. Of course, we're not developing this clinically, but I hope I can convince you there are definitely good therapeutic antibodies in the patient tumors, and we can even find those, right? So at our technology in the company, we call this STAD, which stands for Simultaneous Target Evaluation and Antibody Discovery. Because most of the AI drug discovery companies rely on a pharma partnership or published literature to come up with novel target. And then they design either the small molecule or antibody drug. But in our case, we start from this huge tumor collection, and based on the antibodies, we can tell which target is potentially more interesting. Basically, if the target is present in the tumor, we see the corresponding antibodies. That means this is a really good target. For example, in the zoo situation, the tourists like to take photos of panda. So panda is a good target. It's very photogenic, right? So anytime a zoo has a panda, you see a lot of photos. Whereas goats are not so interesting because it's so boring to take a photo of goat and share it on your social media. So based on the antibody signals, we can already rank the targets. If we see the antibody in the tumor, then we think it's promising. So based on this antibody AI signal, we can tell that PD-1 is better than PD-L1. And unfortunately, we don't see any antibodies against CD47. So it's possible that making antibody against CD47 really don't help the patient. So we don't really see this. And so based on the antibody signal, we already have a ranking of which target are potentially interesting. And then in the second step, we use more functional genomics and big data to help us prioritize the target. When GV20 was started, we were actually a CRISPR screen company because by the way, I was a professor at Dana-Farber Cancer Institute at Harvard for 20 years. And so we were among the earliest to use CRISPR screens for oncology and immuno-oncology studies. And so most of the world that published CRISPR screens also actually use the algorithm developed in our lab to get the hits from the CRISPR screen. So in the company, we also did 3 years of immune-related CRISPR screens. So we look at our own data as well as published CRISPR screens to see whether a gene knockout has phenotype. And then we also look at cell lines, mouse tumors, human tumors, DNA, RNA, protein, epigenetics, and single-cell data, some with immunotherapy data to help us see whether the target is potentially interesting and also generate a hypothesis on the target mechanism. And then with this, we decide, okay, we go after this target, we test so the AI already predicted antibody. So we test the antibody for binding, then we characterize the antibody in vitro and in vivo. And that's how we came to our lead program, which is currently in the phase 1 clinic in the US. So our lead program is a novel innate immune checkpoint that we discovered at the company. So right now for immune checkpoint blockade, the PD-1, PD-L1 is the best antibodies, right? In order for PD-1, PD-L1 antibodies to work, the cancer cell needs to effectively present their mutated peptides onto the cell surface for T-cell receptor to recognize the cancer cell as foreign and kill the cancer cells. Unfortunately, PD-1, PD-L1 only works for a minority of tumors because a majority of the tumors have known antigen presentation defects. So if they're not effectively presenting the antigens well, then the T cells don't have a chance to recognize and kill the cancer cells. And this constitutes both primary and acquired resistance to PD-1, PD-L1. But our our body has a mechanism. Our natural killer cells are supposedly specifically recognize any cells with antigen presentation defects and kill them because they usually indicate the cell is viral infected. And so we wonder in the tumors, why don't NK cells kill these cancer cells with antigen presentation defects? We found that on the cancer cell, there is an overexpression of a ligand called IGSF8. And it has a receptor on natural killer cells and a receptor on dendritic cells to suppress NK and DC. And we could use AI to design a blocking antibody to really block this and activate both NK cells and dendritic cells. So the very first step, we use AI to look at which targets have good antibody signal. We know that the whole B7 family, many of the checkpoint genes, they all belong to this immunoglobulin superfamily. And so we ask, are there other IGSF family members with good antibody signal? And then we saw this gene. We also look at our in-house CRISPR screen data, both in vivo and NK co-culture screens. So normally, when you knock out the IGSF8 gene from cancer cells, it does not influence cell growth in vitro. But when the cancer cell is under immune pressure, whether we grow the cancer cell in the mouse or we co-culture the cancer cell with NK cells, when we knock out this gene, we really see the cancer cell being killed or they grow slower. So we saw, this is kind of immune-related gene. And we look at also this gene's expression pattern across TCGA. So in here, orange are the tumor expression and blue are the normal expression. You can see in many, many solid tumors, IGSF8 is overexpressed. The middle panel includes some cancer types where the normal samples are either not available in TCGA or are too few. So we only draw the normal the tumors, and you can see it's highest in melanoma. And the only two cancer types where IGSF8 is higher than normal compared to tumor are two kidney cancers. So if you work on immuno-oncology, you know that kidney cancers are always opposite from other solid tumors. And so this actually is a good indication that this might be an immune-related molecule. We next look at IGSF8 expression correlation with other genes to give us some idea what it might be doing. And so we see, again, each column is one TCGA cancer type, and each row is another gene. And the color indicates whether this other gene is positively or negatively correlated with IGSF8. If it's blue, it's negative correlation. If it's red, it's positive correlation. And so we ask, okay, what is IGSF8 correlated with? So when IGSF8 is high, the tumor have low CD8 infiltration, low cytolytic activity, low PD-L1. So suggesting these are immune-cold tumors that are unlikely to respond to PD-1. Also, the tumor will have low B2M antigen presentation. And the last four factors are autophagy factors known to degrade MHC class I at the protein level. So when they are highly expressed, the MHC class I might be expressed at the RNA level, but at the protein level, they are not effectively producing or presenting their antigen. And this is when IGSF8 is really high. So normally, IGSF8 is highly expressed in the brain because our neurons never really express MHC class I. So without MHC class I, it suppresses all the adaptive immunity, and highly expressed IGSF8 suppress the innate immunity. And we believe the cancer cell just co-opted this brain mechanism to suppress the innate immunity when they lose their antigen presentation. This gene is actually really conserved, 90% conserved between human and mouse. But using our AI, the antibody we predicted, when we say we de novo predict the antibody against a target, we are just pulling out the antibodies that are already in the patient tumors that we predict to be against this target. And this antibody has similar binding affinity to human, mouse, monkey, and rat, so we can do all the studies with the same Fab. We can see that when we co-culture the cancer cell with natural killer cells, and then we add this antibody, and the NK cells can kill the cancer cells better. We've also tested this in multiple syngeneic tumor models in melanoma, non-small cell lung cancer, colon cancer, and we also tested two different triple-negative breast cancer models. And in each case, we also compare anti-IGSF8 with anti-PD-1. And you can see in each case, this antibody has good monotherapy efficacy and sometimes even better than anti-PD-1. we can also half the dose of each of the antibody to combine them together, and that's the purple line. You can see it's the lowest line in every model. And so we do see good synergy, or at least complementary effect of the two antibodies, because anti-PD-1 is activating the adaptive immunity, and anti-IGSF-8 is activating the innate immunity. We did RNA sequencing of the tumor after the monoclonal antibody treatment. We always see consistent increase in NK-mediated cytolytic activity, increased antigen presentation, increased cell adhesion, and increased T-cell signaling, which is really exciting. We look at the tumors after the monoclonal treatment, and we can see a significant increase in infiltration of NK cells and dendritic cells, and also some increase in the CD4 and the CD8 T cells. And for the B16 tumor, even if we start the treatment after the tumor grow out, with 4 treatments, sometimes the tumor can completely disappear. And if we stop the treatment, the mice can remain tumor-free for even longer time, suggesting that this is kind of a complete responder, which has never been observed for the B16 model with other monoclonal antibody, because B16 has a known class 1 antigen presentation defect. And so PD-1 has some effect, but not very well, but very good. And so this drug, because it's fully human, it has really good developability. We didn't have to do any optimization at the CDMO company, and we get very good yield, good developability. The monkey and rat GLP tox is also very safe. The phase 1 trial is really progressing smoothly in the US clinic since this spring. And if you see how this antibody works, basically, once we add the antibody, the NK cells will kill the cancer cells if they lose their antigen presentation. And that by itself will force the cancer cell to present their class 1 antigen a little better. In addition, the antibody blocks the dendritic cells, so the DC gets more activated, so it's able to present antigen presentation from class 2 also better. And so both of them can activate T cells, and the DC and NK cells can also kind of help each other. So while we do anticipate single agent efficacy and synergy with anti-PD-1, PD-L1. And so far, if you look at the approved checkpoint inhibitors, all of them are on the adaptive immune side. I would say T-cell has been studied to death, right? But so far, innate immunity, people are interested in macrophage, dendritic cell, and NK cell targets. But so far, many of the genes are different between human and mouse. And in human, they're also polymorphic. So, the testing of these antibodies in vitro or actually in vivo are very, very challenging. But in our case, we are able to find this gene which is conserved between human and mouse. And we can use just a monoclonal antibody to activate both NK cells and dendritic cells simultaneously. So we think it has really good potential. And interestingly, for this antibody, before we went to the IND, we only know the NK receptor in human. And then we submitted the paper, and the reviewer said it cannot be this receptor in mouse. We only realized that later. And when we were trying to find the mouse receptor, we found, it also have dendritic cell receptor. And the NK cell receptor, dendritic cell receptor are different, and they're also different between human and mouse. So 4 different receptors. But the antibody against the ligand can block all of the interactions, which I think attests to the power of getting antibodies directly from patient tumors. So it's actually blocking some interactions before we even knew they existed. And so we really kind of are looking forward to this drug in the clinic. Of course, we are not just getting one target. There are additional targets that we're interested in. Some are tumor type specific. Some of them we are getting functional binders, but others are more kind of potential ADC targets. We only have resources to pursue some of them, so we are kind of in partner discussions with pharma. And so in terms of AI drug discovery companies, there are many. We are nowhere even exhaustive. But first of all, many of the companies really cannot come up with novel target. And some of them need existing antibody to do optimization. In our case, also there are companies, they get antibodies from patient tumors, but they usually only take a handful of exceptional responders to get antibodies. In our case, we are getting these antibodies from tens of thousands of samples, right? So, and from AI, we can, one at a time, like produce or predict hundreds of binders against a particular target. And these they seem to be rich in functional binders, and that's how they can repeatedly expand in many patient tumors. And they are fully human, and we have a molecule in the clinic already. So I hope this will convince you that we're really at the intersection between high-throughput genomics, big data AI, and cancer immunology, immunotherapy. In some sense, we are using tens of thousands patients' tumors to do experiments for us, and then using AI to listen to the patient immune system on the insight they are trying to tell us. The first program is a monoclonal antibody, but like nowadays, people are interested in antibody drug conjugate, radioligand therapy, bispecific antibody, or multi-specific antibody, and also cell therapy. And all of them takes the identification of tumor-specific targets and an antibody against it. And I think this is an area that we could potentially come up with novel targets and antibodies. And my ultimate goal is one day if we can profile a patient RNA sample, and then based on the tumor profile, figure out an antibody cocktail that's personalized for that patient, encode that as an RNA cocktail, and deliver this through liquid nanoparticles for the patients to generate the pool. That will be really fantastic. So I want to thank our team. We have most of our headquarters in Cambridge. We also have a research site in Shanghai. I want to thank our advisors and investors, and also more importantly, our clinical sites and patients on the trials. So thank you very much.